things. All right, so last time we talked about the main organs, which is the roots as well as the shoots and the leaves, and we ended up about the flowers, and flowers are actually modified leaves. Modified for what? For sexual reproduction. That's the modification for that. So it's a modified leaves, and then of course we are talking about the Angiosperms, the largest group out of the plants, and it's basically fruits come from the leaves. Before we'll go into fruits, and today at the end we'll talk about the fruits, but let's kind of look at this particular stem. So again, the stem, and we will cover it today in the lab as well. So in this particular case, stem, the place where the leaf is coming out from the stem, known as the node. And then there will be another node, and then another node, another node. Now, the space between the stem, between the leaves known, or between the nodes known as the internode. Inter means in between. Like for example, if I want to fly to Europe, I will be leaving from intercontinental airport between the continents. Same kind of, or if I'm traveling on a high speed between the states, I most likely will be traveling on the interstate, right? So in this particular case, internode means the space of the stem between the nodes. And then of course, each node most likely will have some sort of buds, auxiliary buds, and the terminal or apical bud. And we already discussed that a little bit. So point attaching is the node, internode between the nodes. Now, the arrangement. There is more than 3 major arrangements of the leaves, but we are not in botany. We're barely touching or scratching the surface of botany. But here's the 3 which you need to know. First of all, alternating. Alternating is basically 1 leaf per node. And usually it goes one way into another, into another, into another. Alternating the directions. Why? To maximize the amount of sunlight captured by the leaves, because remember, leaves' major function is photosynthesis. There is of course modified leaves like flowers and the thorns, etc., but generally it's a photosynthesis. So in this particular case, we're looking at the alternate, and that's all sometimes referred to as a spiral, because if you're looking from the top, it will go in a spiral way. So alternating 1 leaf per node. Then there is a 2 leaves per node, known as the opposite, because leaves will be facing in opposite direction. Once again, to maximize the amount of light captured, because well, sunlight equal to food. So please do not forget, and that particular reaction will be on the lab test as well as the lab practical exam, the photosynthesis / cellular respiration. Some of you wrote a perfect chemical parts, carbon dioxide plus water, but you forgot the light. No light, no photosynthesis. All right, because photosynthesis literally means light. And then of course we have something which known as a whorl. It's more than 2 leaves coming out. About the whorl arrangement when we discuss the horsetails. Horsetails usually have several, up to like a half a dozen of tiny little bitty leaves coming out. So in a round kind of formation, whorl. Okay, so let's kind of take a look close by. Alternate arrangements, basically 1 leaf per node, then the opposite 2 leaf per node, and then 3 or more leaves per node. We are talking about whorl. So in this particular case, math and well, in biology is relatively simple. 1 too many. So 1, that's alternate, 2 will be opposite, and more than 2, it's whorl. All right, so here we go. Here's the actual examples: whorl, opposite, and then the alternate, right? Let me kind of take a look. What about this guy? This guy shows the alternating. This guy shows opposite. And I don't think I have another whorl one, but need to include. Now, leaf venations. What the veins are? Veins are something which known as the vascular bundles. This is how the water and minerals coming to the leaf to do the photosynthesis or to process of photosynthesis, and then this is how the sugars produced by the process of photosynthesis will be removed. So those particular ones are known as the leaves, and there is more than 3, but we are going with the 3 major types of venation and the parallel venation. Parallel venation, whenever you're looking at leaf, the veins are running kind of parallel. Yes, from the point of math, it's not parallel because they're starting from kind of one point and then they will end up at the end of the leaf. But from the biology's sake, it is parallel venation, and typically this is monocots. You see parallel venation, most likely you're looking at the monocot. Now, pinnate venation, let's go with the palmate venation. The petiole, the main, that particular kind of stem part of the leaf known as the petiole, whenever the petiole or vascular bundles coming in and they spreading out like a palm of the leaf, palm, right? Goes into coming in and then spreading out. That would be palmate venation. And then whenever you have secondary vein goes into either side, that would be the pinnate venation. Please know the leaves with the different types of venation will be on the lab practical for sure. Those ones are most likely, if you'll see that venation or that venation, most likely you're looking at the dicot. Most likely. There is always exceptions, but most likely you're looking at the dicot. All right. So moving on. Now, here's the thing which some of the students messing up on. We have a leaf. Well, first of all, we have the venations, right? Just the arrangement of the veins within the leaf blade, and it has a parallel pinnate and palmate venation. Now, the arrangement of leaves, especially in the compound leaves, will use the same term, palmate and pinnate, but in this particular case, it's not talking about the arrangement of the veins, but the arrangement of the leaflets. And let me kind of show you momentarily, but make sure that you understand that there is a venation and there is the arrangement of the smaller leaf of the larger leaf, and those are leaves. So in this particular case, they're using the same terms, and it could be a little bit confusing. Hopefully, I'll show you the examples in the lab, and you will be less confused. But yes, botany decided to use the same terms for. So let's kind of go with the leaf types. First, there is more than 2 types, but we'll talk about 2: simple leaf and the compound leaf. What's the difference? Well, kind of obvious, and we're not talking about double, triple compound leaves. So in this particular case, one petiole, see? So we have a stem, the part of the leaf coming out from the stem, the petiole, and then we have a leaf blade. One petiole, one leaf blade, that's a simple leaf. And in this particular case, it would be pinnate venation. Everybody see that? Any questions on that? So we have a simple leaf with a particular venation. One blade, one petiole. Now look at this particular one. One petiole coming out, and then you have leaflets coming out, more than one blade. So now we're looking at the compound leaf. So first, whenever you're looking at the leaf, especially on the lab practical, figure it out, is it a simple leaf or is it compound leaf? And again, the difference is simplified speaking. It's a little bit more complex than that, but we're not in the botany. I don't have time to explain more how they're breaking it down, but more than one leaflet, it means it's a compound leaf. So here we have one blade or leaflet, one leaf on per petiole. More than one leaf per petiole, that's the compound leaf. Now, Here's coming a slightly confusing part. Here in the leaflet, within the leaflet, we see the pinnate venation. Everybody see the pinnate venation, everything is good, right? But now let's take a look at the arrangement of the leaflets. The leaflets coming out, we have a main, the petiole, let's call it petiole right now for simplicity's sake. So we have a main petiole, and then the leaflets coming out on the opposite side. Very similar to the venation, therefore it's called the pinnate arrangement of the leaflets. So it's a pinnate venation and pinnate arrangement of the leaflets. So two different things, venation and the arrangement of the leaflets in the compound leaves. And then of course there will be a palmate venation as well as a palmate arrangement of the leaflets. All right, so here we're looking at the first one. So we have a simple leaf, one blade in one piece, no problem. It could be a very smooth, it could be jagged, it could have all kinds of things. So, but once again, you're looking at the leaf, it's one leaf, one petiole, done. It's a simple leaf, and then all you need to do is identify the venation. In both of those, the venation known as a pinnate, right? All right, moving on. Now, here we have a compound leaves. And in the compound leaves, you have one of the petioles, and it's spreading out into spreading out in several leaflets, clearly separated leaflets. All right. And now let's take a look right here. So simple with a pinnate venation, compound, and you don't really have to think about the odd or even pinnate, just as long as you know the pinnate. In this particular case, compound pinnate tells us the arrangement of the leaflets. If it says compound pinnate, it means that it will be a compound and the leaflets coming out on the opposite sides or going on the opposite sides from each other. In this particular case, you see they kind of coming in and then spreading like a palm. So compound palmate, same kind of idea, coming in, that's one of the petiole, and then it goes into a separate leaflets. But those leaflets are spreading like a palm of your hand, therefore it's a compound palmate. In this particular case, let's take a look on this leaf. It's a compound palmate leaf with the pinnate venation. So again, don't mix and match the venation and the arrangement of the leaves. Arrangement of the veins and arrangement of the leaves, they're using the same term. So in this particular case, compound palmate arrangement of the leaves, and then when you're looking at the venation, it would be pinnate venation. All right. Generally, I'm not trying to trick you on that one during the lab practical. So whenever you will have a question, it will ask you, what is the leaf type? And if it's a simple, you will tell me simple. If it's a compound, you will need to tell me if it's a compound palmate or compound pinnate. If it will ask you, what is the venation? This is where you will need to tell me parallel, pinnate or palmate venation. I try not to mix and match. Like I would not ask, probably would not ask on the lab practical, name the leaf and what's venation is on a specifically compound, because it might get a little bit confusing. Again, don't have to worry about the even and odd, but basically in this particular case, if it's terminating at the only one leaf at the end, it will be odd. If it's a two leaves at the end, it means that it's even. All right, so let's kind of take a look. Palmately compound, pinnate compound. So here we have a petiole, the petiole between the leaflets known as the rachis. Later on today during the lab, we'll cover that. You will see the rachis. It will ask you to show, well, to label the rachis. But rachis is basically a petiole between the leaflets. So here we go, between the leaflets. All right. Again, compound palmate, compound pinnate leaf arrangement. On both of those, the vein arrangement would be pinnate. So this one is a palmately compound leaf with the pinnate venation. So please do not mix and match. All right. Moving on, plants can be subdivided into several major categories. The annuals, they complete the whole life cycle from the seed, essentially, to the seed, from the seed to the grown plant, to the flowers, to the fruits, and then back to the seed in 1 year. And then they usually die, but not necessarily. I had a tomato plant, which is essentially a year annual, but it was surviving for like 2 or 3 years in my backyard until freeze killed it. Yeah, and of course the fungi are not plants. You need to understand that that's not the correct, that's not the picture, should not be here. But generally they survive, and it's not really a surviving, it's from the seed to the seed in 1 year. They're completing the whole reproductive cycle in 1 year. Now, biennials, like the carrots, as well as the most of the flowers, most of the regular flowers, will go probably somewhere around 2 or so years. So the first one is vegetative growth. This is where it's getting ready to spring the flowers to produce seeds, and then the seeds goes on the 2nd year, and usually it dies off. And then if it has more than 2 years, so from the seed to the adult plant and eventually to the seed of the next generation, takes typically 2 years. And then perennials, again, biology type of math, 1, 2, many years, more than 2. So from the seed to the start reproducing could take several years, or could take dozens of years in some cases, but for the trees to grow in and then start producing the seeds. That might take some time, more than 2, and usually they live for several years, produce several generations of the seeds. All right. So typically most of the annuals will not have something which known as the woody tissue, so they are all herbaceous, and some of them are monocots, some of them dicots. And how do I know that? Remember, we're talking about monocots versus dicots, the amount of flower parts. If it's a multiple of 3, 1, 2, 3, 4, 5, 6 petals, it's a monocot. If it's a multiple of 4, 1, 2, 3, 4, or multiples of 5, it's most likely would be a dicot. So here we go again. Angiosperms, major subclasses, monocots versus dicots. 1 cotyledon versus 2 cotyledons. Veins usually parallel, pinnate, or palmate. Vascular bundles, we talked about that last time. Scattered, arranged. Fibrous roots. Usually you have fibrous roots. Kind of hard to tell which one is the main root. Here you definitely have a main or tap root and the secondary roots coming out. And the floral parts usually in the multiples of 3. Floral parts is usually multiples of 5 or 4. All right, so here we go. Monocots, multiples of 3, multiples of 4 or 5. So here we go. Let's kind of see this one. And this is where I need your answers. A monocot or dicot. You can count the petals, or you can count stamens. So this one, monocot or dicot, the yellow one, the lily. It's a tiger lily. D, dicot. Anybody else paying attention to what I'm saying? Dicot. Okay. 2 people. Everybody else absent. All right, multiples of 3 is the monocot, multiples of 4 or 5 is the dicot. Let's count together. 1, 2, 3, 4, 5, 6. 6 is multiple of 3. Now let's count the stamens. 1, 2, 3, 4, 5, 6. Multiples of 3 again. So I have to be a monocot right here. On this one, 1, 2, 3, 4, 5, 6, 7, 8 petals. Multiples of 4, so that one gotta be dicot. All right. And please be a little bit more active whenever I'm asking you. Nothing wrong to answer, nothing bad to answer wrong, because we are practicing, figuring it out what exactly is happening on that particular part. And if you're not answering, you're forcing, you're telling me basically that you're not active. Not active means you are absent from the class. All right, so again, leaf venation, parallel veins will be a monocot, pinnate or palmate most likely will be a dicot. Bundles scattered throughout the stem, that's a monocot. They are arranged in the ring, most likely a dicot. As a matter of fact, most of the trees, like the wooden trees, that's the dicots. And you might heard that the palm trees are not actual trees, and palm trees are monocots. So fibrous root system versus the taproot system, already talked about that. Let's this is the taproot. You see the main root with the secondary roots coming out versus fibrous roots. This is kind of hard to say which one is the actual main root, and that's the onion, of course, and that's the dandelion. So let's see another one. Here we have a picture literally from a backyard. Monocot or dicot? I need everybody's participation right now. You can do it in direct message. You don't have to do it in front of everybody. I need to know, is it a monocot or dicot? Well, guys, what I can tell you, it's wonderful that you can count. 3, multiples of 3 straight away, so gotta be a monocot, right? Yeah, that's wrong. Yes, I know, I tricked you a little bit. Yes, it's not an that's not the exception. No. Remember, multiples of 3 in a flower arrangement. Is that a flower? Do you see a flower anywhere? It's not a flower, it's just the leaves. Bunch of them, by the way. So if you don't see the flower, what are you going for? You're going for a venation. Let's take a look what kind of venation it has. In this particular case, compound leaf. What kind of venation? We have a main vein coming out, and then they spreading, right? Like this. It's a pinnate venation. In order to be a monocot, it have to be a parallel venation. That's a pinnate venation. So most likely, you're actually looking at the dicot. Okay, so please try not to get stuck only 3. 3 of what? Flowers. Now we're looking at the leaves, then like, okay, well, leaves have to look at the venation. So see what's available to you. Use your critical thinking right there. So everybody see that it's a dicot now? Yes? Okay. Yeah, I know I tricked you. I probably will not use that slide for the lab practical. Same kind of idea. No, you don't have flowers. So which one is that? Is this particular plant monocot or dicot? Tell me. That's a dicot. We'll have a really nice palmate venation. You can see the petiole and then the veins spreading out literally like your fingers on the palm, right? So in this particular case, it's a pinnate venation, means non-parallel, If it's a non-parallel, it has to be a dicot. Good job. So don't get tricked by the sneaky professors. Yeah, I'm trying not to trick you. But again, keep in mind that, and in this particular case, this one, is it a simple leaf or compound leaf? Compound. Everybody see it compound, right? So here's the stem. Here's the petiole coming out of the stem. As a matter of fact, you can see that it's on a separate, well, on each side. So in this particular case, the arrangement of leaves would be the opposite. But now we're looking at the compound leaf because it has several leaflets. Technically, it's all compound leaves. But within the, so it would be a pinnate compound leaf with a pinnate venation. Make sense? All right, so keep that in mind. Good idea. Monocot, dicot. Dicot. Okay, cool. Cool. So hopefully you got that, and we'll practice a little bit more in the lab. Meanwhile, I will pull out the second part of today's lecture, which is the flowering plants. And today, this particular part of the lecture, we'll talk about the flowers, and then of course we'll talk about the fruits. So let's kind of take a look on this flower. Took it last spring or spring before. So monocot, dicot. Now we're looking at the flowers. Counting stamens kind of hard because there is a lot of them, but you can clearly see 1, 2, 3, 4, and 5 petals. Just in case, especially if more than one flower available to you, double check, because sometimes the petal may be ripped off. On this one, that's actually a fossil about what, 60, 70 millions of years old. We can kind of see that it has 1, 2, 3 petals. 3 petals versus 5 petals. Dicot, monocot. Even though, once again, it's not even a flower no more. It's fossilized. It's an imprint of the flower. But we can say that most likely we're at some sort of monocot. All right, so reproduction. That, of course, is a sexual reproduction, but we'll start with asexual reproduction. Asexual reproduction versus sexual reproduction, gene exchange versus no gene exchange. Asexual, creating the clones of its own. Genetically, the parent plant would be identical to a daughter plant. Okay? Genetically, identical clones. While in sexual reproduction, because we have a sperm and an egg merging together from presumably different plants, even if it's from the same plant, so-called self-fertilization, the arrangement of the genes will be slightly different because we have a heterozygous, dominant, recessive. As a result, it will, during the meiosis, it will spread a little bit. So anyway, different types of the stems allows for asexual reproduction. Quite often you can literally pinch a little bit of the a part of the plant that particular pinched part, and it will grow into a complete plant. So now you have 2 plants and they are identical genetically. Like, for example, here we have a mass of ginger. If I'll cut a part of that, and it's a stem, underground stem of ginger, if I'll cut that and I will plant it, it will grow separate. So again, ginger, stem, rhizomes, and we can do the cut. Same thing with the potatoes. I can cut potatoes, let's say in 4, as long as it has something which is known as the eye of the potato, or essentially it's the bud. It will grow into a separate plant. It could be 2 or 3 different plants coming out from the same pieces of the, well, from the different pieces of the same potato. So in this particular case, it will be asexual reproduction. Then strawberries, of course, if you ever grow the strawberries, you will have the main plant of the strawberries, and then it will send something which is known as the runners. That's the stems which will later on touch the ground, and then it will grow another plant. Both of those will be genetically identical. This is how you ended up with the patches of strawberries. So here we go. This is what typically strawberry does. We have that particular plant, the main plant, a runner or stolon, and then it will get into the ground, will start producing its own root, and eventually it will grow into exactly the same plant. There is another way, which actually we are using. It's a similar idea. In this particular case, a lower branch can be put down underground, and eventually it will create its own plant. Later on, it can be separated. There will be a root system for that, and you will end up with the two trees which are genetically identical. Humans are also using a very interesting way known as a grafting. By grafting, we're talking about you have a stalk plant, usually it's a woody plant, and then you can graft, you can cut and then plant on top of it a scion of a different plant. Now you'll have the genetically different one, even though it's considered to be asexual reproduction. Friend of mine who is in botany, well, actually plant physiology, but it's part of botany. He had the apple tree in his backyard. And what he did, he's like, well, apple tree, a fairly nice apple tree. So he cut one of the branches of the apple tree and he grafted a pear. So now you have apples on one side, and on the grafting branch, you will have a pears. So technically it's an apple tree now with the apples and pears. But he didn't stop there. I think he grafted something else on that particular tree. I think it was, I might be wrong. He had, let's say he had a red delicious. Here we go. I remember it now. So he had a red delicious tree, and then he got the grafted the pears, and then he grafted on another branch. He grafted the yellow delicious. So now he has it's still an apple, but it's a different variety of the apple. He has a red delicious from the same tree. He can do the red delicious, golden delicious, and the pears. Later on, he played with the citruses. He did the lemon. He has a lemon tree, but it's inside lemon tree, which he grafted kumquats and I think the oranges. So he has a same tree producing three different fruits, so to speak. But it's still considered, even though once again the genes of those will be different, considered to be asexual reproduction. Micropropagation, we're usually using the sterile environment. We're growing them in a medium. Basically, it's agarose with the minerals and water. As a result, you will end up with the seedlings. Oh, you're putting the seed and then seedlings which are being cultivated in these sterile conditions. This is how we are making or producing lots of different variety of the plants and then selling them as the plants in the stores. For example, orchids. Orchids produce that way. You have the seeds of the orchids, you're planting them into literally a test tubes, and then they start growing, and then you're planting them into the sphagnum and delivering it to the stores. So here we're looking at the lily. Now that's a red lily, and in this particular case, monocot or dicot. Now we're looking at the flowers. So now we need to count the arrangement, and most of you are correct. That's a monocot. Yes, 1, 2, 3, 4, 5, 6. Six petals. That's most likely a monocot. And again, I'll try not to put any confusing images there, and I'll talk about the confusion during the lab. So again, alternation of generations, everybody remember that. We have the diploid, the sporophyte, and then of course the haploid, the gametophyte. Gametophytes are now, especially in the flowering plants reduce drastically. We're talking about handful of cells. That's it. Handful of cells, 6, 7 cells. For the female, 7 cells technically, with 8 nuclei, and 2 or 3 cells for the male gametophyte. Microscopic. They are totally dependent on the gametophyte, on the sporophyte. Sporophyte, the diploid state, now has the predominant form. So whenever you're looking at the tree, you're looking at the sporophyte. In order to find the gametophyte, it will be deep inside of the tree. All right, so now moving on. Here's the parts. Parts of the flower, the whole flower or complete flower usually have 4 major parts known as the calyx, corolla, androecium, ginoseum. Calyx, that's the combination of the sepals. Sepals are usually green parts of the flower. The outside, think about as the whorls, right? So the outside whorl, and usually they are protecting the flower while it's in the bud. And in some cases, the sepals can fall off when the flower is open up. Then we have a showy, typically a showy, bright colored petals. Why we need them? They are attracting any kind of pollinators. And transfer of the pollen, remember, they need to get the pollen from one flower to another somehow. It could be wind, or it could be a pollinator employing some sort of animals to do that. And that would be a mutualistic relationship. I'll give you this, you'll give me that. So what exactly happened? Let's say a bee coming in, getting the nectar, something sweet, right? And usually nectar will be located at the receptacle or near the receptacle. So it have to go all the way down to lick that particular or suck that particular nectar. But during that particular time, the anthers will shower that animal, a bee, with the pollen. Read the animal or plant sperm, right? And then the bee goes to the next flower and inevitably will transfer some of the pollen to the receptacle, oh, not receptacle, stigma of the flower. So here's we have a cross-pollination. So that's the petals, a combination of the petals known as the corolla. Then we have androecium. Andros means male, so male part of the flower, which basically the stamens. Stamens will be made out of 2 things known as the filament and anther. Anther, this is where the meiosis is taking place. And of course, from this is where the male gametophytes will be formed, the pollen. And then the central part known as the ginoseum, that's the female part of the flower, will have a major parts known as the carpel, sometimes referred to as the pistil, and has several parts. We'll discuss that, but the main idea is it will contain ovule, the whole thing, right here, ovule. Within the ovary, later on, ovary will turn into what? The ovary will turn into a fruit, where within the ovule you will have the egg, and egg will be fertilized by the sperm, will turn into the embryo. And also, well, egg usually right here. And then it will have something which is known as a central cell, which will be fertilized by the second sperm, and it will turn into a food source for the embryo, seed essentially. So here what we're looking at, male and female parts. So here we're looking at the male, known as the stamens, anther producing pollen, filament taking the anthers away from the flower. So if some sort of insects will be coming in, or any other animals, they will be taking out the pollen, or sometimes anthers the filaments will take the anthers out, so the wind can pick it up, the pollen, and transmit it throughout. And then of course within the pistil or carpel, it's the same thing. Has a 3 major parts: stigma, style, and ovary, and we'll discuss that momentarily. So here we have on the female part, right? We already talked about the. So Ginoseum, pistil or carpel, the entire portion of the female flower, and its central part, it has the upper part, which is usually sticky. This is where the pollen will land on and will get recognized. Oh yeah, that's the pollen from the same species. So it will get okay to grow. And then there will be a special cell inside of the pollen, which is known as the cell tube. It will literally grow into the tube. It will grow through the style, and then it will go into the ovule. Within the tube, the sperms will start swimming in toward the egg and toward the central cell to create the seed. All right. So now here we have the ovary. Within the ovary, there is an ovule. So in this case, we have one ovary, one ovule. How many seeds it will be able to produce? Only one per fruit. Think about the apricot. You're looking at the apricot or peach or avocado, all right, or plum. Anyway, you're looking at that, you're opening it up, the ovary, the fleshy part, and then you see only one seed. So here we go. That was one ovary, one ovule. With one egg, therefore only one seed. Something which is known as a simple fruits, and we'll talk about that today a little bit more. Okay, but at this particular time, make sure that you understand the whole thing is the ovary, the part inside known as the ovule. It has a many ovule. If the ovary has more than one ovule, then there is a possibility to have more than one seed within the fruit. So here we have the stigma, the sticky part of them. Then there is the style, same kind of idea as the. It's not stylus, it's a style. It's basically a connection of the stigma to the ovule. So the landing of the pollen, and then the tube grows, and the sperm coming and fertilizing the egg. And then of course we'll have the style, support the stalk, moving the stigma out, so it will be facing whatever the pollen coming in. And then we have the ovary, becomes fruit and contain one or more ovules. And depending if it's more than one ovule, it will have more than one seed within the fruit, because ovary will become part of the fruit at least. And we're not going into the complex fruits. There is some of the fruits which are becoming, well, part of the receptacles becoming a fruit. We're not going into that, don't have time to discuss all of those parts. We'll go in a very simplified version of the botany. So, and now ovule. Each ovule will contain only one egg. Make sure that you understand that. Ovary can contain many ovules, but each ovule will contain only one egg, which will be fertilized and which will develop into a seed. And remember, a seed will contain three major parts: the cover, right? And then it will have the embryo and the food source. And today we'll talk about how it's produced, specifically the embryo and the food source within the double fertilization. So male parts of the flower, we're talking about the stamen, and stamen will be made out of two parts: the stalk, so to speak, the filament, which is taking out the anther, and within the anther, this is where the pollen is produced. Pollen produced, and again, look, in this particular case, show a very large, bright red petals. So in this particular case, let's say a bee coming in, while it's trying to get inside to get the nectar, it will be literally showered by the pollen. And then later on, when it goes into a different flower, it will inevitably touch the sticky part. So the pollen will be transferred cross fertilization. So anthers producing pollen, and filament supports the anther to take the pollen out, so it will not be inside of the flower. So here we're looking at the white lily. You can see that particular, that's the stigma right here. This is a part of the style. And you clearly see the anthers, and that particular yellow stuff produced is nothing else but the pollen. Can they self-fertilize? Yes, many of the plants can do that, but they prefer, since it's a sexual reproduction, to have the cross-fertilization. Petals are colorful. In most of the, well, in many cases, they are colorful, especially to attract the pollinators. Many of the plants, however, still using just the wind. And if it's a wind pollinated plant, it usually does not have a very showy, colorful petals. But petals are generally kind of to attract, so any kind of pollinators, it could be insects, or it could be birds, or could be bats, for example. So mammals, birds, as well as the insects are general pollinators. So in this particular case, it's just showing, hey, I'm here, see me, and therefore you will get some sort of food. But while it's getting food, look at those particular tiny little bitty yellow spots. What is it? It's basically all pollen. So they are using, well, the plants are using animals to transfer the pollen from one to another. So all petals known as the corolla, already talked about that. And then the sepals, usually green parts, to protect the flower at the bud stage. In many cases, you will not see them, and we'll talk about incomplete flowers. So here we are looking at the petals, and then the back of the flower, of course, is the sepals. Now, receptacle is basically a place where the ovary sits on. And we're not really talking, you don't have to worry much about the superior, inferior, or halfway in. We don't have time, it's botany. You definitely will talk about that in the botany class, and I think Central Campus offers botany and zoology. But in this particular case, if its ovule sits on top of the receptacles, it's known as a superior. If the receptacle is kind of right here, then it would be known as the inferior. And if it's encased in the inferior ovary, then we'll talk about the receptacles will become part of the fruit, but we're not going too deep. It will be very confusing. So here, well, again, looking at the flower, a complete flower, meaning it will have all four major parts. All of the sepals, well, we'll have the calyx, we'll have the something which known as a corolla, all of the petals, and then it will have the male as well as the female parts. That's a complete flower. Now, let's kind of take a look on the flower. Yeah, perfect flowers will contain both male and female parts. In the same flower, you'll find the part which producing pollen, and the one which will produce the ovules or eggs. Imperfect would be either a male flower or female flower, either producing only pollen or either having the eggs produced. And that happens to some of the plants. Like, for example, you most likely heard about corn, most likely ate corn, right? We here in the South, we definitely, corn is a part of our diet. Flowers in the corn, they are not very showy because they are wind pollinated, but typically you will have near the stem of the corn plant, you will have the female flowers, and then on the top you will have a male flowers. Male flowers produce the pollen. It will be blowing off by the wind and will be accepted by the pistillate flowers, which later on will turn into a corn, which we all know and love, right? So staminate and pistillate flowers may be on the same plant and in different plants, and this is where we start talking about or repeating stuff from the Gymnosperms. We're talking about monoecious and dioecious. Remember that? Monoecious, staminate and pistillate will be located on the same plant. For example, the corn. Corn will have, corn is a monoecious flowers, meaning the flowers will be located on the different parts of the plant, but on the same plant. Usually the staminates will be on top. The pistillates, the female flowers, will be kind of closer to the middle part. This is what will grow into the corn themselves. And then, of course, dioecious. Staminate and pistillate will be located at the different plants. We have a male plant versus a female plant. For example, the willow trees. See, here we have the male flowers producing tons of pollen. And here we're looking at the female flowers, which does not have a single staminate, but that would be having a tons of receptacles. Well, not receptacles, stigmas to get the pollen on. So complete flowers contain all of the parts, all four, right? So we have perfect and imperfect. Perfect will contain both staminate and pistillate parts, so reproductive parts, and a complete flower will contain sepals, petals, stamen, and pistil. As soon as it will become missing one or more, it will definitely be incomplete flower. So let's say if it's a male flower, so it's missing the pistils, therefore it's incomplete. Or a female flower, it will be missing the stamens for sure. So it will be incomplete. So if it's missing one or more, it's incomplete flowers. Now, look at this part right here. We're looking at the sepals, petals. Now we're looking at the staminate and we're looking at the pistil. And in this particular case, look how many ovules it has. It has 6 ovules. So potentially that particular fruit will have 6 seeds. Why I'm saying potentially? Because not necessarily that all 6 ovules will be, or all of the 6 eggs within the 6 ovules will be fertilized. Let's say it was, I don't know, a rainy day or rainy week. So the bees were able to transfer only 4 pollen grains, and therefore only 4 of them will be fertilized, therefore only 4 seeds will form. Make sense? In this particular case, we're looking at the imperfect flower. Why? Because it's missing the stamens. So as soon as it's missing one, it's a first incomplete, and then it will be not perfect, imperfect. Then there is something which is known as the composite flowers. Composite flowers is a very interesting adaptation of the plants. As a matter of fact, there is a whole group of plants known as the composite plants, the composita. Those ones have a tiny little bitty flowers mixed together kind of in a very large flower, a composite flower. So think about the daisy, for example, or if you ever seen the sunflower, we have a bunch of sunflower flowers here. So in this case, each one of those tiny little bitty dots within the daisy or sunflower is the separate flower, which can be fertilized, and it will produce the seed. That's why whenever you're looking at the sunflower seed, sunflower flower, after it has been fertilized, it will contain tons of sunflower seeds. Yummy, right? So anyway, here what we're looking at we don't have to semi and further, don't have to worry about that, but in this particular case, sepals, fused petals, and then you will see the yellow one with the stamens, and then of course the pistil. So it's the complete flower. Everything is fine. Here we're looking at the several flowers on the same stalk, and here we're looking at each one of those dots. Each one of those is a separate flower but combined all together. And we'll talk about the monoecious part versus dioecious. So this is the male flowers, this is the female flowers of the corn. They are located typically on the same plant, and usually the males ones will be on the very top, and the female ones, which later on will give us the corn cobs, would be kind of at the middle part of the plant. And here we're looking at the common arrowhead. There is such a thing, and here we're looking at the male, not only flower, but male plant producing only male flowers, and female plants, so dioecious. Let's take a look on this. Monocot or dicot? Mono. How can you tell? Well, first of all, you're counting petals. 1, 2, 3. Just double checking. Maybe I did something tricky, and you can counting these the stamens. 1, 2, 3, 4, 5, 6. And just in case you're looking at the leaves are given to you, and you can see that the veins run parallel. So at multiple of 3 for the flower, and it's a parallel venation for the leaves. It gotta be a monocot. Yep. Thank you, guys. Yeah, you kind of getting it. All right, so let's kind of keep going. The life cycle. Again, we talk about the production of the gametophytes, and the gametophytes will be produced by meiosis, of course, within the flower. So that would be the gametophytes, and the male gametophyte at the adult stage will have either 2 or 3 different books going differently about what considered to be a mature male gametophyte. It could be either 2 cells, or one of those particular cells later on will split into 2 sperms, and we'll talk about that momentarily. And then it will talking about the whole gametophyte will have 1, 2, 3 cells. On a female gametophyte, which will be located deep in the ovary, we'll end up with a megaspore. Megaspore will start dividing. We'll talk about that. It's a little bit more complex than the males. And then we'll end up with the 7 cells. 1, 2, 3, 1, 2, 3, and 1. So 3, 3, and 1. 7 cells with 8 nuclei. Yes, I know females, plant females are complicated, right? So let's kind of talk about the male meiosis first. Meiosis, which will produce from the sporophyte, a gametophyte. And it's straightforward. It's a typical meiosis without any kind of problems. We have the microspore, which is a diploid, goes through the meiosis 1, it goes through the prophase, metaphase, anaphase, separating the cells, everything is going good. And then it goes into a meiosis 2, creating the 4 cells, known as the tetrad, haploid. Now everything is great. Each one of those particular cells of the tetrad will produce a single pollen haploid grain. All good. Everything is fine. And of course, the pollen tube, pollen grain will have a special protection. Why? It will have additional layers. Don't have to worry much about that. But in this particular case, the layers will protect the cells inside cell, and then later on cells, in order to, well, from the drying out. So pollen grain here, we have a single cell gametophyte, and then it will, through the meiosis, it will split into 2 cells. So that already considered to be a 2-cell male gametophyte for the flowering plants. Later on, there will be another division, mitosis, because it's already haploid, and one of those particular cells will split into 2, and they will form sperms. Remember, each pollen grain carries 2 sperm. They are essential for something which is known as the double fertilization for the flowering plants. And double fertilization for the flowering plants will be on the test. I'll show you the basics, and you need to be able to recall that basic steps from the double fertilization on the test to get the full credit. And of course, I'm assigning partial if you went somewhere out there. And I had a student a couple of years ago in the face-to-face class. She wrote literally like 3 or 4 pages of double fertilization in plants, saying something like that. I don't remember double fertilization in plants, but here's my take on it. And she goes into quite interesting, but completely wrong, but very creative ideas. It's like, you know, 50 Shades of Grey summary in the plant style. So it was strange. Let's put it this way, more or less school appropriate. In some cases, it wasn't. on the borderline, but it's creative. It was creative. I was able to give her partial credit for the creativity. And I went to my department head. I'm like, here, read this. And she's like, what's that? And I'm like, double fertilization in plants. She's like, oh, this is what you are studying? I'm like, no, it's not alone her, but that was creativity. I'm like, I'm giving her credit. She's like, yeah, I can see why you're giving her credit. So again, male meiosis, straightforward, no issues, no problems. Again, as typical meiosis, we're starting with the diploid microspore mother cells, which undergo 2 divisions of the meiosis, form 4 microspores. Remember, spores are haploid. Each of those spores will first divide through the mitosis into 2 cells, considered to be a mature pollen grain with the 2, known as the generative cells, which will split into 2 sperms, and one of them is a nucleus tube cell. Or tube cell, basically that particular cell, whenever it will end up on the pollen, will grow into the style in order to lead those particular 2 sperm to the egg. So here we have mature pollen grain, which is the male gametophyte, a mature male gametophyte, and now you can count it has only 3 cells. Mature male gametophyte in the flowering plants, 3 cells, 2 to 3 cells tops. And this is different types of the pollen. Pollen has a different shapes, has a different receptors on the top. As a result, the plant knows which particular pollen is its own, so to speak. And we're looking at the, well, technically it's immature pollen grain, but we have the pollen tube cell right here. And then we have the generative nucleus, which will split, or splitting, you can see, splitting into 2 sperm. So whenever the pollen will end up on the proper pistil, this is what's going to happen. The tube cell will start growing into a tube-looking structure, leading to the egg. And remember, we have 2 sperms, so sperms will start swimming. It's happening inside of the flower now, swimming through that particular tube to reach the egg. Yay, fertilization. So here we have the pollen grain, microphotograph of pollen grain, the tube, and this is sperm number 1, sperm number 2. 2 sperms are essential, okay? 2 sperm, very important. Now, female meiosis. Female meiosis is a little bit more complex, okay? And in this particular case, what we'll start with standard meiosis. Everything goes from the mother, microspore or megaspore goes into a 4, well, that double division of the meiosis, producing 4 megaspores. That's the typical meiosis. Everything is good, but it will stop there. The typical will stop. Out of those 4 megaspores, 3 of them will die off. Only 1 will survive. 3 will literally disintegrate, break down. Why? Because the large one, the one which is survived, will get all of the nutrients. So instead of meiosis in the female will produce out of, well, from 1 cell, instead of 4, it will produce 1, but very large cell comparatively to the sperm, for example, or to the male gametophyte. So a megaspore stays. Now, the megaspore, within the megaspore, the nucleus will undergo 3 rounds of mitosis. So we have 1 cell on the first mitosis, it will end up with the 2 nuclei without splitting up. 2 nuclei, then the second round will give us 4 nuclei, and the third round will take 8 nuclei. So 1 cell, 8 nuclei. See, getting complex. So we still have the megaspore with 8 nuclei, and then it will go through the process of cytokinesis, formation of the cell. And typically, when typical cytokinesis happens, you will end up with 1 nucleus in each cell. So out of 8 nuclei, you're supposed to get 8 cells. That's not what's happening. Yay! Instead of 8 cells, it will end up with 7 cells. Because one of the cells will take 2 nuclei. Okay? And this is what's showing right here. We have 1 cell, 2 cells, 3 cells, 4, 5, 6. Each one of them contain 1 nucleus. And then one more cell, which is kind of located in the middle of the ovule. This is the thing known as the ovule. And all of those 7 cells is actually female gametophyte. And that particular central cell has 2 nuclei. That's important. So let's kind of summarize all that. We have the meiosis will end up producing only one very large megaspore. Within the megaspore, the nucleus undergo 3 mitotical divisions. 3 mitotical divisions will produce 8 nuclei, and then cytokinesis will produce 7 cells. Out of those 7 cells, 2 are extremely important. They're all important, but 2 are extremely important for the double fertilization, and we will focus our attention on those 2. One of them will be located right and next to the opening into the ovule known as the egg. Right next to the egg, there will be 2 cells which are known as the synergids. They're kind of helping the egg. Then we have a central cell. 2 of those cells are essential in the double fertilization. Remember, whenever the pollen tube grows, it will end up right here. It will release 2 sperms. So egg and the central cell. Yes, antipodal cells, 3 antipodal cells are important as well. Don't have time to talk about that, sorry. If you're really interested, you can read it on your own or take the botany class. Very, very interesting, but I'm not a botanist. I will tell you right away, but the plants are not my most favorite subjects to learn in biology. Anyway, so in this particular case, since our time is kind of limited, let's kind of focus on the central cell and the egg. Egg is the haploid, has a haploid nucleus. It's only 1 nucleus. All right. Central cell will have a haploid nuclei, but there is 2 of them. So what exactly happening during the double fertilization? The pollen tube grows, carrying 2 sperms. So whenever it will reach the polar or micropolar end, basically the opening, the sperm will go inside of the female gametophyte. And the first sperm will fertilize the egg. So we'll end up with a diploid cell, a zygote. Everything is good. But remember, we talk about the double fertilization. Why double fertilization? Because there is a second sperm. Second sperm coming in and the egg is already fertilized, closed for business, done. It's a zygote now. It's the beginning of the sporophyte generation. And the sperm is like, well, I'm simplifying, of course. It's not how it is. Like, well, I travel all the way here from a different plant, from a different flower, was carried by the bee, and now there is nothing to fertilize. No, I'm here to fertilize someone. And it's like, oh, you, the central cell, you look like a good substitution. So the second sperm will go and fertilize, will merge with the central cell. Now the central cell will have a third nuclei. Eventually, whenever it will merge together, it will become triploid. So the double fertilization will result in a diploid zygote and a triploid cell. Diploid zygote will develop into the embryo. Triploid cell will develop into something which is known as the endosperm, the food source for the embryo. Make sense? So next time, if you're eating, let's say, almonds or pistachios, you're essentially eating the food source for the embryos. We are horrible to the plants. We're eating the embryos, and then we're eating, well, we're saying, we'll eat your food source embryo, and in a way, we'll eat you as well, because you're so small and you can run out. And by the way, endosperm tastes good, right? Yes. Or for sunflower seeds. or hazelnuts or pecans. I have a pecans, and they start growing, and the squirrels start visiting more and more often. So this is the generally double fertilization of the plants. So here's again female meiosis, starting with the megaspore, goes through the meiosis, 4 megaspores. Out of 4, 3 of them will disintegrate, 1 will end up going through 3 cycles of mitosis, creating 8 nuclei but 7 cells. And that's basically the whole female gametophyte. That's of course inside of the ovule, and ovule will be inside of the ovary. So pollination is basically transfer of the pollen, and you can see all kinds of pollinators right here. The bees, insects are quite common. You're looking at the birds. We have the hummingbirds around here, and in some areas, especially tropical ones, the bats can do that. And we can also artificially plant, transfer the pollen if needed. This is one of the largest flowers ever, the corpse flower. It called corpse flower because instead of nice and sweet smell, it's producing the very, very bad stench, because it's attracting the flies as well as the corpse beetles. They're coming in, they're looking around, trying to lay eggs. There is no rotten meat, and therefore they're moving on. But in this particular case, they transfer the pollen. And as you can see, it was about like, that picture from the Smithsonian Museum, couple of about 10 years ago now. They had one of them blooming, and it was extremely large, as you can see, about 6 feet tall, and the smell was unbelievable. The stench, it was like you almost want to vomit there. And some of the people who were standing in line to watch it, they actually were kind of got sick. Anyway, so pollination and fertilization. Remember, we have the transfer of the pollen is a pollination. As soon as it being recognized, it will the pollen tube will start growing, carrying 2 sperms. Those particular sperms will get, and they will fertilize the egg, and then it will fertilize the central cell. As a result, we'll end up with diploid zygote and the triploid endosperm. Make sense? I'll simplify it for you. So again, same kind of thing, showing a pollen tube carrying 2 sperms, and the first sperm will fertilize the egg. The second one will fertilize the central cell, creating a diploid zygote and triploid cell, which will develop into the endosperm, a food source. So here's what you need to know for the double fertilization. I simplified for you as much as I could. All right, well, let's kind of take a look. We have 1 sperm, which is a haploid, will merge with 1 egg, which is also haploid. It will create a zygote, which is a diploid, which will develop into the embryo. So far, so good? Simple enough. So double fertilization, 1 egg and 1 polar nuclei will be fertilized by 2 sperms. Sperm number 1, or first sperm, will fertilize the egg. Haploid will create a diploid zygote, which will develop into the embryo. But it's a double fertilization. So second sperm is still haploid, now will fertilize central cell, which is a dikaryotic. Dikaryotic means has 2 nuclei, each one of them haploid. And now it will end up as the triploid endosperm, which is a stored food to nourish the diploid embryo. If you'll write me this or in that particular format, I'll be happy. I am happy. I'll give you the full grade. Again, haploid sperm fertilizing haploid egg will create a diploid zygote, which will develop into the embryo, also diploid. That's now as sporophyte generation, gametophyte, well, gametes actually, and the sporophyte generation. Now, second sperm will fertilize the central cell or two polar nuclei, which are dikaryotic, and will create triploid endosperm, which will develop into a food to nourish the embryo. And quite often we're going for that particular endosperm. Any kind of nuts we're eating, or almost, we are going for the endosperm. And in a way, we're eating the embryos as well. So here's what we have. Pollen grain lands, then it grows. You can see the two sperm going in, and then we ended up with one sperm, well, one fusion sperm and egg will give you what? Embryo, diploid, don't forget that. And then fusion of one sperm and the two polar nuclei will produce endosperm. So here we have pretty much the same thing, again, showing you the pollen tube. And now we're looking at the ovary with several ovules. So to fertilize each of those ovules, we'll have to end up with the different pollens, because here the two cells, two sperms will be used up, so to speak, in this particular ovule. Within the ovary, several ovules. Each ovule will contain one egg. So here what we have, same kind of idea. Now, early embryo development. Whenever the embryo development, and this is where it's talking about the dicots and the dicots, monocots. Whenever it develops, what the cot means, cotyledon. See right there, cotyledon. Cotyledons are embryonic, essentially they are embryonic leaves. Embryonic leaves which allows or helps the embryo to digest the stored materials, because again, stored materials of the endosperm will be in the compact form, in the form of starch, in the form of proteins, in the form of fats. Those ones needs to be broken down. The starches have to be broken down into the simple sugars, proteins into amino acids, the fats into fatty acids, etc. And this is what cotyledon, here we have two of those, and here we have only one cotyledon, will do. So say that's, let's say, a kernel of corn, lots of starch comes in, the cotyledon breaks it apart, and then feeding the embryo so embryo can grow. Within the embryo, there will be several major parts which will develop into different parts of the seedling. In this particular case, make sure you understand what cotyledons are. Now we're looking at the epicortals. Epicortals will become the actual, not embryonic, but the actual leaves. Then the hypocotyl will turn into, it's essentially embryonic stem, so it will turn into the actual stem. And then the radicle will turn into the actual root. That's it. Both of the corn, well, both of the monocots and dicots have pretty much the same structure of the embryo, and we'll talk about that, not this, but next lab on Thursday. So here we have the fruit development, and we need to go talk a little bit about the fruits. So here, see, ovary, one ovary contained one ovule. One ovule will create one seed. Yes, I know I'm a little bit over time, but bear with me, we're almost done. So the fruits can be subdivided into several categories. And let's kind of split it into a very simple. First of all, there is a fleshy fruits. They have a flesh, kind of soft tissue. And the dry fruits. Simplified speaking, I'm oversimplifying in this particular case. All of the dry fruits will be simple fruits. So we're looking at the walnuts, we're looking at the almonds, hazelnuts. So all of those, if it's dry, it's automatically simple. Not exactly that simple, but let's keep it for your and my sake. on that level of botany. If it's a dry, it's automatically simple fruit. No problem. They will be subdivided. The dry fruits, we'll talk a little bit more about that. We'll talk about dehiscent, indehiscent, etc. But if it's a dry, it's automatically simple, and then you will need to figure it out if it's dehiscent or indehiscent. Now, if it's a fleshy fruit, means it has flesh, it has, well, what we are considered to be a fruit, right? Like an apple or pineapple or the blackberries, strawberry, raspberry, peach, and so on. In this particular case, they can be simple, they can be aggregate, and they can be multiple. Don't have to worry about accessory on this particular level of botany. So if it's a fleshy, soft, then it could be simple, aggregate, and multiple. And I'll give you the examples of what each one of means. So simple fruit. Simple fruit means that we have one flower, everybody say one flower, one ovary. It could be one or several ovules. So it might be a number of seeds. If it's a one seed, it's automatically simple fruit, moving on. Flesh is simple, done. But in this particular case, it might have several seeds because it was one ovary with several ovules, and those particular ovules has been fertilized, therefore you will end up with several seeds. So, but in this particular case, look, one flower, one ovary, one or several seeds, simple fruit, flesh. Now, the aggregate, one flower, but now several ovaries. Therefore, several seeds will be produced during that, well, during the development of that particular fruit. And that known as the aggregate. One flower, several ovaries. And example, well, a good example would be a raspberry. Strawberry will kind of put aggregate fruit as well. Now, multiple fruits. Multiple fruits, now we're looking at the multiple flowers, multiple ovaries, multiple seeds. Pineapple would be a great example. Also, we'll talk about the figs. I live in Friendswood. Friendswood started as the fig farm, right? So figs, as well as there's something which known as a mulberry. Not many people know what mulberry is, even though I've seen some trees of the mulberries around here. So types of the fruits, don't have to worry about that. Don't have to worry about the berry, pepto, hesperidium. Don't have time to go why exactly banana is not really a berry. Well, banana is technically a berry, but it's a false berry. Blueberry is not a blueberry. It should be called scientifically blue false berry. Cranberry is not a berry, technically speaking. It's a false berry. However, look at this particular one. Tomato is a berry. It's not a vegetable, it's a berry. So next time you're eating berries, tomato is. Same thing with the eggplant. You can say, well, it's a vegetable. Yes, vegetable and berries or fruits is different in the common language versus the scientific language. So in this particular case, eggplant, pineapple, or not pineapple, a watermelon, pumpkin, they're all true berries. See right here, melon, watermelon, something which known as a peppo. And then most of the citruses as hesperidium, don't really have to worry about that as long as you understand that this is simple fruits. Then there is the aggregate fruit, and then there is the multiple fruit. For simplicity's sake, we'll say apple, peach, cherries will be referred as the simple fruits. Strawberry is the aggregate fruit. Sunflower seed, it's a dry, and it would be a simple fruit. Beans or peas, for simplicity's sake, we'll say it's a simple fruit. So, again, whenever we're looking at the fruits, we're looking, is it fleshy or is it dry? If it's a dry, it can be two possible options, dehiscent and indehiscent. How you can tell them apart? Dehiscent, whenever the fruit is ready to release the seed, it will open up and the seeds will be released. Crepe myrtles, you know that usually they have a very pretty flowers, they're blooming throughout the year, pink, white, etc. They have a fruit, and it's a dehiscent fruit whenever it's like a capsule, and whenever it's ready to release the seed, it will open up, the seeds coming out. That's the dry dehiscent. Dehiscent, the fruit itself will open up to release the seeds. Indehiscent, on the other hand, will stay as the fruit without releasing the seed, meaning, let's say, acorn, right? Whenever it will fall, it will not open up to release the acorn itself. It will stay as the shell inside. And that would be indehiscent. This is what you will need to know about the dry fruits and be able to identify. Now, on the fleshy, you will need to identify if it's a simple one or if it's an aggregate versus multiple. And I'll give you the examples, and those particular examples will be on the lab practical. So here we go. Several ovules, several seeds, one flower. Could be one or several ovules. So don't have to worry about that, but this is what separates, or this is how the part of the endocarp, mesocarp, and exocarp, which is a part of the fruit, separates the why it's considered to be a berry, why it's considered to be a hesperidium, why it's considered to be a pepo, etc. And here's, I think there's, yeah, here we go. True berry, the pericarp is fleshy throughout, and here we have a seeds. The hesperidium, we have a leathery rind, and then the pepo, thick and inseparable rind. So, but you don't have to worry about that, as long as you understand that it's a fleshy fruit and it's simple, and we're moving on. Oops, sorry, here we go. So yeah, drupes, again, fleshy, one seed, you see one seed, that's automatically fleshy. You see one seed, it's automatically simple. All you need to do is figure it out, is it the fleshy one or is it the dry one? Coconut is unusual drupe. It's not actually not that particular husk of the coconut is actual fruit. We're going for the seed itself. So dry fruit, already talk about the dehiscent, like the peanuts, they will open up. Tamarind, right? Open up, releasing the seed. Right now, if you're going south of Houston, like on Highway 59 or so, you will see they probably already got most of the cotton fields. Cotton is the dehiscent. It has a capsule which will open up, releasing that particular white stuff. Inside of it will have a tiny little bitty black seed. And then indehiscent one. Indehiscent, like corn. You know, whenever you're eating corn, there is a transparent kind of, well, the seed is covered by the transparent thing, and that one is the actual fruit. It's not, well, it's a capsule. It's not releasing the seed. Same thing with the acorn or hazelnuts. That one stays. The seed is inside of it and it's not being released. The embryo will have to literally break it apart whenever it starts growing. Yeah, you don't have to worry about Samara, acne, or anything like that. Now, aggregate fruits. Now, fruit derived from several pistils in the same flower. Strawberry, raspberry, blackberry, all of those particular things. So here we're looking at the single flower, but now we're looking at the several pistils, and those pistils creating the overall fruit. So many of those, yeah, here we go, the blackberries, right? And then the multiple fruits are derived from many pistils within the many flower, and you have a pineapple, fig, and that's a mulberry right here. More of that particular stuff. And well, basically, we already talked about that, so don't really have to worry about that. So we have the seed coat, then the cotyledons, and then we're looking at the embryo, the embryonic leaves, the embryonic stem, the embryonic root, right? And I think we talk about that. Radicals develop into the roots, then plumule develops into the leaves, and then hypocotyl develops into the stem. And we'll talk about that during the lab next time. Yeah, that's pretty much all we need to see. You'll see that particular picture, cotyledon, a single endosperm, and now you're looking at the plumule, hypocotyl, and then the radicle. And this is what essentially you will see under the microscope. Endosperm, that one is the cotyledon, and you can say a single one, and that will develop into the leaf. So, and that one develop into the stem, and that develop into the roots. So same thing with the dicot seed, but we'll cover that. I'll stop right about now because we'll cover it in the lab number 10. So stop recording.